So to keep going, I wish to invite to the podium Professor Gordan Lauc, and you, I guess, know him from previous conferences, but that's not the reason that he's invited. He is among the world's foremost experts on glycosylation. And please, Professor Lauc.
So good morning. Thank you for being here. So now I will switch from the forensic medicine to personalized medicine, which is the other part of this conference. And I will try to introduce you to the topic of glycans, which is, I know, not really familiar to many of you. And I will tell you something what you have learned through our National Center of Excellence in Personalized Healthcare in the last decade, and how we are trying to translate this knowledge through a company called Glycan Age, which is dedicated for commercialization of glycan assays. And what epidemiology has taught us, that longevity is a decision. We actually decide whether we are going to have a long and healthy life or not. And we can even do it late in life. So if in our 50s we have 4 healthy habits, we can expect 10 more healthy years of life than if we do not have these healthy habits. So we can actually decide to live healthier. But that decision is not easy because we don't really know what are the healthy habits for each of us. With all the advance of modern science, the only thing which we know that works for everybody is not eating, calorie restriction. And it works in humans, it works in animals. So this monkey here has been hungry for 20 years. And by everything we can measure, he looks younger than his brother who was eating normally. But he does not look happy. I'm not sure that not eating and being hungry for the rest of our lives is something we really want to do. But if we want to make a decision about healthy diet, we really don't know what is a healthy diet for me. This is why we have thousands of books preaching different diets, because each of these diets will probably help some of us. But there is no healthy diet which will help all of us. And I think one of the good examples is lactase persistence. So we are raising cattle for more than 10,000 years. Eating milk was essential for survival. And approximately 7,000 years ago, mutations appeared which enabled persistence of the lactase activity. And nobody in the north of Europe survived without that gene. So in the north of Europe, everybody has lactase persistence, but 2/3 of the planet do not have it. So even for this essential mutation, which is decided between life and death, the majority of population was not selected to have it in over 7,000 years. So now if you think about all the more recent changes in our diet, the abundance of food, the abundance of carbohydrates, we are simply not genetically adapted to this change, and it will take thousands of years for us to become adapted to these new patterns of eating. And the similar is for physical activity. Although the absence of physical activity is definitely not healthy, it's one of the main causes of diseases. Many people overexercise. Actually, especially middle-aged people, when they start to exercise, they usually over-exercise and do themselves more damage. So even physical activity has to be personalized. And one of the main healthcare challenges today is that we don't know. Because everything we know is based on a standard human. So we take 1,000 people, measure something, take an average, and declare this is a standard human, this is what works for standard human. But there is no standard human. The only thing which works for everybody is a caloric restriction. So we know generally it's sleep, it's diet, it's stress, it's physical activity, but what is important for me? Would it be more important to sleep 1 hour more, or maybe spend that hour in the gym, or skip lunch? We don't know that. And we need biomarkers to tell us which of these interventions actually help me. But unfortunately, despite all the technical success, genomics still did not fulfill expectations. We have very little usable data in our genome. I sequenced my whole genome 5, 6 years ago. I have no use of it. I don't know what actually to change based on my genetic information. We have to do much more research. But even after doing much more research, I'm not sure that we will become much smarter, because only maybe 10 to 15% of longevity is genetic. Only up to 30% of complex diseases is genetic. And when we think about the way life functions, the polymorphisms, the differences in a single letter in the genome, have to go through the multiple layers of interactions before the final phenotype. So it's really not easy to predict a phenotype based on polymorphisms. And I think we can see very clearly from Manfred's presentation, some things are doable, but some things are really difficult. And not everything is written in the genome. And I think where we actually have to look, we have to look in this ultimate omic layer, which are the glycans. Glycans integrate genetic, epigenetic, and environmental factors. If you look at this protein revolving in the middle, everything hairy on its surface are glycans. We cannot understand the function of that protein without looking into its glycans. This is the S-glycoprotein of the SARS-CoV-2 virus. Or we think about antibodies. Antibodies are the main weapons of our immune system. FAB part is binding to an antigen. But what to do next is deciding by the FC part, actually deciding by the glycan in the FC part. And if we have glycans like this, the IgG will be pro-inflammatory. It will kill the target. So it will be an enemy, kill an enemy. But we can put a different glycan. And this glycan will tell antibodies, this antigen is a friend. We do not want to attack it. Actually, we will suppress inflammation and let that antigen live. And this is a decision made by a glycan. An analogy I like to make are these two birds. So this is a polypeptide defined by our genome. This is a glycoprotein with a peptide and a glycan part. And we can be the best scientists in the world. We can have all the funding. We can have a perfectly planned experiment. We can execute it perfectly. But if we are trying to understand flight, and we are using this bird as a model, the closest to understanding flight we could come is to see this bird walking. Because bird without its feathers cannot fly, protein without its glycans cannot perform all the functions protein is supposed to perform. And the majority of proteins are glycosylated. Before I go on, one word of caution. Glycation is something completely different. Glycation is a chemical reaction between glucose and amino groups on proteins. We usually measure it as HbA1c. We know it's very important, but it has nothing to do with glycosylation. Glycosylation is an additional layer of complexity. So the proteins will have a polypeptide part defined by a gene. It's a billions of years of wisdom. And then it will have a glycan part defined through a network of dozens of hundreds of genes. This is the network we mapped for IgG glycosylation, which will create a glycan structure, which will be added to the protein. So if we look from a functional aspect, alternative glycosylation is analogous to coding mutation. We all know coding mutation can kill you. Wrong amino acid, wrong protein sequence, protein does not function. Alternative glycosylation can have a very same effect. But from informational or evolutional aspect, glycan composition is inherited as a complex trait. So this enables diversity without the risk for genetic heritage. And I think we provided a very good demonstration that this is really the way I'm saying, is by analyzing the collaborative cross-cohort of mice generated by Grant Morahan in the Western Australia. So he started from the 8 founder strains, genetically very diverse, and then crossed them for 3 generations, and then inbred the cousins. So he created more or less the clones of cousins. So you can analyze them. And then when we analyzed all those strains, some of the strains separated completely from the rest of the population. So their glycan composition structure of their immunoglobulins, function of their immune system was completely different. And there were no mutations here. It's just reshuffling of the allelic variants. So the 3 generations of interbreeding generated stable and heritable differences in IgG structure between these mouse strains. So it's a very powerful evolutionary tool. And over a decade ago, Vlatko and I speculated, we published a perspective article that glycans are the 3rd revolution in evolution. And I think we are close to proving that. This hasn't been published yet, but we have shown that the peak of glycoprotein evolution was happening when all the processes needed for multicellularity were being developed. So sometime in evolution, when we needed to become multicellular, it's glycans who came into play and enabled it. And also when we look at the individual level, glycans are what makes us different. Even blood groups are chemically glycans. So these are 6 random individuals, different color, different glycan. People are different. We are all not the same. Glycans are a big part of it by genetic, epigenetic, and environmental factors which contribute. So glycans integrate all these informational elements into chemical structures which can be reliably quantified. And this is what we do, and this is what I'm trying to persuade most of you. Let's join us and let's do it together. Because alternative glycosylation can have very dramatic effects of protein function. So most of you know about rituximab. It's one of the biggest best-selling drugs in the history. And it has one tiny problem. It has this tiny little core fucose, which is inhibiting its activity by over 99%. So we are giving a blockbuster drug for over 20 years, which is 99% inhibited by wrong glycan. And a couple of years ago, blutuximab was developed, exactly the same antibody, but with proper glycosylation, and it's potent in 100 or 500 times lower concentration. And this is where big pharma is moving at the moment, doing a glycoengineering, making drugs which actually work because they're not inhibited by having a wrong glycan. And the reason why you don't know much about it is that glycan analysis is difficult. So these are complicated structures. You can have multiple glycans on a single polypeptide. Different glycans can be attached to the same glycosylation site. It requires many chemical steps to create a data, and it's not easy to create the reliable data. And we were the first who went in that direction. We did our first large glycan study 15 years ago. And so far, we analyzed over 200,000 people in collaboration with some of the leading researchers all around the world. Recent review in a chemical reviews revealed that we generated approximately 80% of the global high-throughput glycomic output. So more or less, if you want to do a large study anywhere in the world, you have to come to us. So one of the first things we did, we linked glycans with the genes. That's easy. Everybody has genetic data, so we did the famous GWASs. We are doing them for 15 years now. And this is, for example, the network of genes which regulate IgG glycosylation. The orange ones are enzymes. So the enzymes which actually do the work are not the key regulatory elements. They are the blue-collar workers. White-collar workers are these guys here making decisions. And these decision-making genes are different for different proteins. For example, we did a parallel GWAS for IgG and transferrin. They have exactly the same glycan structures. They have the same enzymes making these glycan structures, but the decision-making genes will be different for transferrin and for IgG. And we did much more work on IgG, and for IgG, we mapped the pleiotropy of these genes, and there are 94 different phenotypes which we identified to show pleiotropy with IgG glycom. So the genes which regulate IgG glycosylation will be predictors of lupus, which is expected, type 1 diabetes, which is expected, but also the cholesterol concentration, HDL, Parkinson, some forms of dementia, things we don't know how they actually function. Another large set of work we did is looking at changes with glycans in people through time. And these are the effects of aging, 5 different cohorts, different glycans. We more or less see the same pattern. Blue are men, we age linearly. Red are women, there is acceleration around the perimenopause age. And when we published that over 10 years ago, the editors of the Gerontology Journal asked, are glycans the holy grail of the biomarkers of aging? And I hope in the next 15 minutes I will convince you that they actually are. For example, when we looked all around the world, 27 populations from different parts of the world, changes in glycans strongly associated with the expected lifespan. So if you live in a country with a longer lifespan, your glycans change slowly toward old glycans than if you live in a poor country. This part is not published, but we also show that at the individual level, glycans predict mortality. So when you look at your glycans in a cross-sectional way, they will predict when you actually died, because we have a cohort which were now followed up for 30 years. And now we are doing 2 replications, and we hope we'll publish this soon. For example, older glycan was a predictor of a severe COVID. Acceleration in glycan aging was predictor of mortality in COVID. So people who gained 5, 10, 15 glycan years within a few weeks in COVID most probably died, but people who did not change, they did not die. We don't fully understand all the mechanisms because, for example, when we looked at the Down syndrome, and Down is accelerated aging, actually people with Down syndrome do not have accelerated aging. They have an acceleration of aging during early life, don't know exactly when, but after puberty, they have exactly the same pace of aging as the non-Down people. And through some genetic identification, we speculated it's one of the kinases, DARK1A, A1A, and that it's unrepaired DNA damage, which could be the driver. And another data from the HIV patients on therapy suggests this might be the case because people who have virus controlled by the antiretroviral drug actually have the same pattern of accelerated aging. The acceleration of glycan aging and epigenetic aging does not correlate. So the glycan age and epigenetic age correlates, of course, because they both correlate with age. But when we look at acceleration, there is nearly no acceleration. This study was published by the Edinburgh group a couple of years ago. We are just doing a replication. Again, we don't see any correlation. When we looked at all the different diseases we could look at, and we did analyze 200,000 people, so we have many different disease cohorts, in many diseases, we see acceleration of glycan aging, and the changes in glycans are the same as we see in aging. So a healthy, older person will have a similar glycan to the younger person with different diseases. And in many of these diseases, we see changes in glycans up to a decade before disease is actually diagnosed. We have very strong papers in diabetes, in prediction of cardiovascular events, and many other diseases where we actually see glycans changing up to a decade before people develop a disease. And we know that these glycans are functional regulators of low-grade systemic inflammation. There are many papers from other people showing that these glycans actually directly modulate inflammation. So the idea we have is that kind of a trajectory from a healthy state to some kind of a complex diseases, and most of the complex diseases of today have inflammation as an underlying mechanism. People usually go for help here. When something is not functioning, they feel pain, they ask for help, they want a pill, and then they become lifelong customers of the big pharma. But what we would like to see, we would like to see these changes very early, when people are not sick, when you just see the changes in glycans, which will lead to disease. And then you can identify these changes, and you can do some kind of preventive intervention, and by suppressing this systemic inflammation, you can maybe postpone or even completely prevent disease development. And this is how and why I developed this glycan clock of aging, which is a functional effector of inflammation, which we can quantify reliably, and which is today commercially available in over 1,000 different clinics. And of course, I have to repeat here, I'm the co-inventor, I'm the conflicted here, of course. You should look at the data and not just take my conclusions as a kind of granted. But if you look into data which other people did, for example, this was a cohort done in Qatar, where colleagues from Weill Cornell analyzed over 6,000 different molecular traits. They measured more or less everything which can be measured from blood in a person. And then when they looked for correlation with age, 9 out of the 20 best correlations were immunoglobulin glycans. And the others were mostly sex hormones. Or the studies we did in the field of cardiovascular diseases, initially we did a cross-sectional study together with Tim Spector and Jim Wilson from UK. We showed that IgG glycan composition correlates with the cardiovascular disease risk score. So there's a correlation between disease risk and the glycan composition today. We used the data to persuade the people from the EPIC cohort, and EPIC was collected 30 years ago. They have a clinical follow-up, so we analyzed the sample from 30 years ago, and we know who actually had a heart attack and stroke in the subsequent follow-up period. And glycans plus age in men were nearly as predictive as the entire AHA score. In women, they were even more predictive. So there is something in the glycans which is telling you whether you're going to have a heart attack or stroke in the future. Again, it was published well, but it's just one study. We went for another application. So the same thing again. Again, it's a sex stratified, different glycans in men and women. And actually, the hazard ratios were similar, around 0.7 per standard deviation. Well, went for another 2 replication. This was published in Circulation Research a couple of months ago. Again, nearly the same hazard ratio for women. So actually, we do see something in glycans predicting heart attack and stroke. If you look at the total plasma glycom, it's predictive of diabetes. So as people are approaching, actually, this was just insulin resistance here, from a healthy state, this is 2 to 4 years, 5 to 7 years, 8 to 10 years. There is a continuous change of glycom from a healthy state to diabetes. The prediction is not so good, but actually diabetes is not a disease. Diabetes is a symptom. And when we went to stratify it, there is a subgroup of people with diabetes with a specific mechanism, and I don't have time to go into details, which we can nearly perfectly predict from the glycom composition today, with the area under curve 0.95, which is nearly perfect. And interestingly, the same glycans which are predictive future diabetes are predicting the response, postprandial response to lipids and glucose. So glycans somehow affect proteins in the membrane, which then associates with the response to the food we are eating. And the best of all is if we change our lifestyle, we can actually revert these changes. So the changes from healthy to diabetes state can go in the opposite direction if we start to introduce healthy life habits. And I think the key question we all have is aging something we can revert? Can we slow it down? Can we prevent it at least a bit? At least can we prevent diseases? And we have shown that the number of interventions which are considered to be healthy, like a physical activity, diet interventions, hormone replacement, can actually improve your glycom and push it kind of toward the backward aging clock. For example, if you do bariatric surgery, people lose up to 30 years within the 6 months. Bariatric surgery is a little bit too drastic for many people, but if you do a caloric restriction to the hungry monkey, yes, it works in most of everybody. This is something like 8 cohorts all around Europe. We had the same effect everywhere. And this was a 2 months of 800 calories a day. That's a torture. We also did a small cohort in US where people were on a low-calorie diet for 2 years, and you can see prolonged effects even after 2 years. But as I said at the beginning, being hungry is not a solution. So there was a study in Europe where they tried 5 different diets. And after the caloric restriction, they put people on 5 different diets. And on each of the diets, some people got better, some people got worse. So there is no magic diet. We have to see which diet actually works for me. Good news is that it's never too late. In the 30s, in the 40s, in the 50s, if you start losing weight, you get in a kind of reversal of aging. Of course, this is good for people who are overweight. Skinny people, if they lose weight, that's not good. Of course, there are drugs. So infliximab works. If you take anti-inflammatory drug, you can suppress your inflammation and change your glycom, but this is not something you want to do preventively. Some other strange interventions work. For example, IVIG works, of course. IVIG is an anti-inflammatory therapy. You give young immunoglobulins to people, and the sialylated glycans suppress inflammation. Plasma exchange works, but it takes some time. So there are no effects after 3 months. You see clear effects after 6 months. And this was a sham plasma exchange showing no effects. And in all these studies, it's extremely important to have a placebo group, because without a placebo group, we can see effects of more or less everything. So we tested many so-called magic anti-aging interventions. One of them was metformin. Even I tried metformin at one point because some of my friends convinced me that I should try it. It's a magic anti-aging drug. And when we did a properly controlled trial, less than 10% of people actually have beneficial effects of metformin. So if you're healthy, don't take metformin just as a prevention. But if you're a diabetic, of course, metformin will help many more people. So we have seen metformin decreasing biological age in diabetic people, approximately 50% of people. The same goes for SGLT2 inhibitors and GLP-1 agonists. So different drugs can have very strong effects in diabetics. Of course, different people will respond to different drugs. Something cool we published earlier this year was a strange study done in India. We just did a glycan analysis there. They used exosome fraction from a newborn pig liver, injected into rats, and rats actually lived longer. And when we looked at the glycan age, and Steve Horvath did the epigenetic age, there was a rapid reduction of both the glycan and epigenetic age. So there is something in exosomes which send information to the body to change biological age. There may be supplements, not yet on the market, but maybe in the future people will develop supplements. For example, the MANAC, N-acetylmanoxamine, seems to be very effective in mice. So you can protect obese mice for hypertension. Don't know whether it works in humans. This is a circulation study, so it's supposed to be a good study, but just one study so far. And I think it's very important to have tools to measure these things, and I believe glycans can really be helpful here. And Cell Perspective paper published a couple of months ago by the Biomarkers of Aging Consortium clearly identified glycans as something which is already commercially used to track responses to lifestyle interventions and which associate with multiple diseases. So the idea is, let's try to use it to actually help individuals. And this is me tracking myself through time. My glycan age is horrible, I'm not talking about it. But what I want to show here is how accurate and precise the measurement is. So this is not one batch analysis. This is every few months I send a sample to the analysis. Okay, I do send it from lab. I just go from my office to the lab. But you see the changes are minor, and then you can even identify small things. I identified the cryptic root canal infection, which none of the biochemical markers was identifying, but something was going wrong. I was looking into it, and then I managed to fix it. And when you look at the numbers are really reliable, which is one of the main problems of all other biomarkers of aging. You just do the test 3 times to get the 3 different numbers. One of the famous colleagues who's also doing this regularly is Mike Snyder. So we analyzed over, I believe, 200 of his own samples. And you can see kind of process of aging. This is over 10 years. But you see 2 age reversals, very effective, done at the beginning, at the end of the study. This was actually published just yesterday in Geroscience. Another famous individual, Tim Spector, he actually responded to metformin. So he's one of the few people who had a beneficial effect of metformin. Or Rob ter Horst is a colleague from Austria testing everything, so we also tested glycans for him. And then you can see changes where in one period of life, he would have kind of a cardioprotective effect in one type of glycan, but more pro-inflammatory effects in others. So you can go kind of deeper and try to understand what is actually going on here. And I think I used my time, so I have just to acknowledge all the funding agencies we have. Managed to get over 40 million euros to analyze glycans, and I have to say thank you, mostly to European Union. And I do have a large group of people who are working, so I can enjoy the beautiful conference all around the world. Although approximately 20 of them are here, and we do have a booth there, so if you're interested, you can find them there. And I thank you all for your attention.